Morning. It's wonderful to be back in Madison, especially on such a joyous occasion. I graduated in 1984 and have had the privilege of coming back here every 3 or 4 years or so. Today, of course, is special because we are here to celebrate Ed Lightfoot's 90th birthday. And I'm pleased to see so many friends in the audience that I have now literally known for decades, and see them develop very successful careers. Many of them because of Ed Lightfoot's mentorship and wisdom, and that certainly includes me. So, as I think all of us know, Ed Lightfoot does not like to be praised in public or in private, for that matter. And we were encouraged, rather than to do that, to speak about what we learned in graduate school and what was of enduring value, in addition to maybe talking about some of our scientific material. So I'm going to take the first 10, 15 minutes to talk about my experience in graduate school here, what I learned, and what I took away from it, and what endured. And we'll start from the beginning. In the beginning of time, for me, and Ed was here, not too far away, down on the square. Probably in 1979, when I'm an incoming graduate student here, 22 years old. And Ed is parting with some wisdom there. It took me only 15, 20 minutes of time with him to decide that he was such an interesting and wise man that I felt that I had to go and join his lab and have him mentor me through my PhD. The gentleman on the left was my sponsor at the Rotary Club. I was lucky enough to have a Rotary Fellowship here for my first year. Now, in one of my first classes, I took, in one of my first semesters here, I took a class on design, and it included doing an econometric analysis of the intermediate chemical industry. And this was my introduction to systems, science, and analysis, and it has had quite an enduring effect on me. So what we are looking at here is the intermediate chemical industry with the fossil fuel-based feedstocks, and then the products coming out, and going from left to right, the prices go up, and the volumes go down. And our task was, in 1981, to figure out if any fermentation products could be introduced into this series of chemical transformations in an economically feasible way. Of course, back then it was mostly alcohols and acids, natural products, and the answer to the analysis was that this was not going to work economically at the time. But as I'll talk about later, things have changed quite a bit in 30 years or so, and this question is now being readdressed. Now, so I learned a few things from this paper, obviously, linear programming, thinking industry-wide, thinking systematically, and so forth. But as you notice, this paper ended up in Science. So this was my term project in this class, and Ed got his hands on the paper and rewrote it in such a clear and elegant way that it was accepted into Science, essentially without review. Now, as you know, I learned from that quite a few things too. As those of you know that have dealt with Ed, he speaks in very complete and clear sentences, and not only that, he speaks in very clear and coherent paragraphs. And at times, if you just record him and transcribe it, you would basically have complete text. And sometimes he would do this at a rapid rate, and it was hard for foreign students to absorb it. And at the time, we coined a unit to describe his speech rate, which was PPS, paragraphs per second. And I swear, at times that he approached one on that scale. I believe Brady Lemhoff has the honor of coining that term. Now, life goes on. So 30 years later, I am now working with Amar Shah, who is with us here today, who is also a student of Ed Lightfoot, and Magnus, who was a student of Dale Rudd's, which is the lab that this model came from. And today we are still doing these simulations. And there is still this question lingering, can we make chemicals through biomanufacturing? And the answer is that this is becoming much more feasible than it was some time ago. So we've now built this model, and this have just predicted price trends of various commodity chemicals over the last 12 months. And you can see These industry-wide models do quite well for many of them, these green curves. Here's one where there was complete failure of prediction. And if you look at the market for that particular chemical, well, there was a big plant explosion, and the demand, the supply changed completely, so the model is way off. And in some of these other boxes, there are cases of dumping that disturb the natural economics, and apparently some executives are under prosecution in those cases. So these econometric models are quite interesting and useful for many purposes. Now, one of the things that Ed Lightfoot always told me was that young people see the future, and always encouraged us to think big and move ahead with big ideas. And based on this systems model and experience, I decided to become what's known today as systems biologist. And here I am about a year later, probably 23 years old, trying to develop systems biology. And the tools of the trade haven't changed so much qualitatively, although quantitatively they have. So we had a GUI that was an engineering paper, maybe size A3 or something like that. That we would draw the basic maps we were interested in, and then we would have transparent paper that we would lay on top of that and draw that in different colors and shift them back and forth to try to learn things. We had the memory stick, which was a magnetic tape that went on the PDP 1155 in our computer room. I had the programmable calculator, Texas Instruments 59, that could do all sorts of cosine functions, hyperbolic ones, and whatnot. Very impressive. And then symbolic computation was done with a number 2 pencil and algebra by hand. So the predictions for me at that time were you can't compute biology, this was professional suicide, and a dead-end career. But as Ed said, young people see the future, and the future I saw then apparently did materialize, and we now have genome-scale systems biology practiced around the country and around the world, as I'll talk about in a moment. Now, as a chemical engineer, well-versed in building complex models based on physical-chemical principles, we all believed that we could build rigorous kinetic descriptions of cell functions and use them to predict and simulate accurately what cells do. And this came down to writing a bunch of differential equations that had to be analyzed. So here's one that's been linearized to get a Jacobian. Jacobian can go through a similarity transformation and produce the so-called modes. And this is a table of modes, each one of them moving on a different time scale. And we would try to understand what these numbers meant in terms of the qualitative dynamics. And we would look at things like this. There is 111, mode moving on the order of 2.5 days, and turns out this is a pool of adenosines, and there were some other structures in tables like this one. This is, for instance, the pool of high-energy phosphate bonds in glycolysis. What I did discover in doing this was that you could change the kinetic constants quite a bit in the model, which are the hardest numbers to get, and this structure remained relatively invariant. And that brings me to the second set of concepts and lessons I learned in graduate school that had a durable effect on my career. That's the notion of timescale separation. And I have here some clippings from Ed Lightfoot's notes, class notes from that time. So here is how he taught us about the Peclet number. It's a flow down a channel that can either be dominated by diffusion if the flow is so slow. If the flow is fast enough, diffusion is uninteresting. So you have a convection and a diffusion limited regime here, which basically comes down to understanding the timescales of these two processes. He also taught us that the structures of systems also can be coarse-grained based on timescale separation. So when he was studying the liver or the kidney and so forth in pharmacokinetics, you could either idealize them as a mixed tank, complete mixing, or as a tubular reactor where there is no mixing. So these are two complete extremes. And if the timescale separation is sufficient between the response time of the organ and the body, the response curves of the output to the input here becomes very, very similar for these two limits. And this was just fascinating to me that you could simplify problems so easily based on timescale separation. And of course, this came down to the interpretation of the dimensionless numbers we use in transport phenomena. But more importantly for me, I actually used this kind of analysis to generate very valuable IP that was issued in the 90s that led to improving viral delivery to target cells for transformation and gene therapy, how to design bioreactors and feed them the right way to mimic in vivo conditions to grow, for instance, bone marrow cells and so forth. So these simple ideas, these simple lessons turned out to be extremely important to me. Not only in terms of design, but also to build realistic descriptions of how cells actually function. So cells don't behave like clockwork. You cannot really describe them by detailed mechanistic models. You can, but if you want to describe real biological phenomena, it's tough. That comes down, of course, to diversity, the problem with defining boundary conditions and initial conditions, and also, of course, that living systems change with time because the genetic code changes with generations, and a model suitable for one generation may not apply to the next. So these are very fundamental challenges associated with doing this. And I, at some point shortly after I left Madison to join the University of Michigan, was thinking about this problem. And I realized that the Jacobian matrix can be decomposed into two matrices. One is the stoichiometric matrix, which represents the structure of the network, kind of similar to those examples that I gave. And G is a matrix filled with kinetic constants or derivatives of rate laws. And this is where all the problem comes from, is to find these numbers and their disparate orders of magnitude. So looking, and we knew that this would always be very, very hard to get, whereas S was something that was easy to get. And in fact, the structure of the modal matrix that I showed before is primarily derived from S, and order of magnitude information about the elements of G. So this led me actually to the most important question of my career that really had a big impact on it. And that was the question, what can we learn from stoichiometry alone and only approximate information about the kinetics? So to make a long story short, this actually worked quite well. This is from the paper from the early 90s. That was the first biological prediction of a large-scale network model. This is a fed batch fermentation here. And these solid curves are just computed curves, and here's the data overlaid on them. So these are pure predictions. Not perfect, of course, but surprisingly good. And the student that did this, Amit Varma, made this comment after his thesis. It's amazing how informative mass and energy balances is if they're properly formulated and actually put in the context of what biological objectives are. So here's Amit, came to California some years ago to help us consult for a company I had started maybe 7-8 years ago. Now here are some bold images that we put out at that time, and this is something that Ed always encouraged us to do. Be bold, think big, and think deep into the future. And we would draw diagrams like this. So based on what I said, it turns out that the stoichiometric structure of a network actually confines the solution, actually described as a polytope. We knew that if we had the kinetics and regulation, we could shrink that to a smaller set of solutions. But as I said, there is distal causation in biology that changes those parameters. So evolution will actually make you move around inside that polytope. And I'll show you now data later on that shows how that works. So we started working on concepts like this one, where you had solution spaces associated with matrices that describe the capabilities of an organism, the space of all phenotypes, and a particular point in that space was an expressed phenotype of a cell. And in this way, we could actually combine both physical-chemical causation in biology as well as distal causation that represents evolution. And this is now called dual causation, and genome-scale models can now describe dual causation in biology. Now, so those are some of the technical lessons I learned as a graduate student that had an impact on my career, but we learned other things. We learned how to work long hours, especially in the computer room, overnight, where we learned numerics, computations, and algorithms. And we were, of course, always joking in the wee hours of the morning, and there were some unforgettable phrases that came up. And this one here comes from Jim Rawlings, immer, and das Computer Zimmer. For those of you who know German, that is actually quite funny. Back in those days, PhD students had to learn a foreign language And obviously, Jim was taking German. So after 12 credits of German, we got jewels like this from him. So that's a summary of some of my durable lessons from graduate school. A very memorable and joyful part of my life. What I would like to do now is to maybe talk more about my career in a broader perspective and how these sorts of lessons allowed me to build an academic career that is now 32 years in duration. And what I did end up wanting to focus on was to build what are now known as genome-scale models based on all the capabilities encoded on a genome that can be contextualized into what are called whole cell models that describe the function of a cell in a particular environment. So if I have a few minutes here, it's maybe fun to look at this image for a while. So this is a bacterial cell. This is a rendering of E. coli, and we all have our own strains of E. coli growing in our gut. We don't only have our personalized genome, but we also have personalized E. coli in our gut and other bacteria living on us. So let's look at this a little bit. So the volume of that cell is on the order of cubic microns. The dimensions here are about microns or so. There is a DNA string in there, yellow in color, which is about 5 million base pairs, maybe 5.5 million base pairs in natural strains. There are maybe 5,000 genetic elements or so. In blue, we see the protein, so that's the proteome. About 2.5 million protein molecules in a bacteria. There are maybe 2,500 genes expressed in E. coli, so you ratio those two numbers, and you get, on average, about 1,000 copies of every protein in E. coli. They vary quite a bit in their distribution. There are elongation factors associated with protein translation that are maybe 200,000 in number, so almost 10% of the proteome. Then there are others that are in just a handful of copies that may have very specific regulatory effects. So there's a multi-scale problem there, just like the time-scale problem that I mentioned. The purple dots in there are the ribosomes that make the protein, and there are maybe 10,000, 15,000 of them in a growing cell. All of this is packed into this volume. And then, of course, the little metabolites floating around, maybe, 2,000 or so different ones, are smaller and float around. All of this has to be coordinated with exquisite accuracy to get a cell to execute its physiological functions and survive in the wild. Today, we can profile these components in great details. We sequence whole genomes. I don't know if Fred Blattner is here or not, but he sequenced the first E. coli strains in 1997, pathogenic strain in 2000. So we have this information now in mass. We can now do deep coverage, quantitative coverage of the proteome, and so forth. So we have a lot of information about these cells in terms of their molecular profiling. We can also now go into the genomes and edit them. You've all heard about genome editing. We can go edit bacterial cells. We can now edit human cells. So that's a whole new locus of technologies that has developed to a remarkable degree of sophistication. And now I'm going to show you that we can actually describe these things now quantitatively with systems models that cover maybe not all, but maybe up to 80% of the mass of the components you're looking at in there. Now, bacteria live in us and on us. As you know, it's often stated that we have 10 times more bacterial cells than human cells. That turns not out to be quite true. If you look into the January 8th issue of Nature this year, you find out that if you include red cells and platelets, it's about equal count. It's maybe 8 to 10 or so, but some people don't view those cells as real cells because they don't have a nucleus. But anyway, these cells play a big role in our physiology. For instance, the sterile human body is apparently odorless. All odor created of the human body comes from bacterial activity. So we talk now about microbiomes, skin microbiomes, oral microbiomes, gut microbiomes, and so forth. Bacteria also are extremely important in agriculture and ag bio, and I don't know if you've heard some of the more recent developments there, namely that people are spraying seeds now with bacteria before they go into the ground, so they form the right root microbiome that allows the plant to access the nutrients in the ground much more effectively. And people involved in field trials tell me that the results are absolutely astonishing in this regard. So bacteria are quite important to us everyday life. And in terms of evolution, they've been on the planet for more than 3.5 billion years. Eukaryotes were maybe a little over 1.5 billion. We maybe for a few million years. So they were here long before us, and they will also be here long after we are gone. So these are interesting things to study. So now I'm going to take you through a little bit of material, not too much detail, on how we build systems models of biological functions with the ultimate goal of trying to relate how all these molecular components and their properties are organized to produce coherent biological functions. So early on, this was easy to do for metabolism. When the first genomes came out in the late 90s and were annotated, we could pick out the metabolic genes, and those are the genes that catalyzed the pathways in metabolism. And we could organize that information into proper chemical reactions, as shown over here for glycolysis, write pathway maps, and we could do this on genome scale. That results in a few thousand of these reactions, maybe a couple of thousand for E. coli. What we can then do is to take that information, these chemical equations, and they can be converted into mathematical equations. And so now we have mathematical representation of all this knowledge. And here we show a stoichiometric matrix, no big surprise, where the rows are the compounds and the columns are the reactions, nodes and links if you're a systems biologist. Turns out, as you saw in the previous images, that if you try to solve network-based equations, say in a steady state, with these equations, you don't get a unique solution, but you get a solution space, which says that the bacteria has a choice in which metabolic state it chooses to implement. And how do we know which one it would choose? Well, nothing in biology makes sense except in the light of evolution. It's a famous quote. So if you do understand the evolutionary background of a cell, you might be able to guess which phenotypic state it should pick. And of course, we were dealing with laboratory strains that are growing rapidly, developed over decades to grow on minimal media or complex media. And one would expect that perhaps they would have evolved to grow as fast as possible. So you can actually search these solution spaces for the fastest growth state and generate results like the ones shown here, where this is the oxygen uptake rate, malate uptake rate in this case, and on the z-axis here is the output or the cellular growth. And this multicolored polytope here is that solution space, and the best growth solutions lie on this edge here based on an optimization problem, optimization computation, constraint-based optimization type computation. And in many cases, the data lies very close to that edge. And therefore, we have succeeded by 3 simple ingredients to predict phenotypic states from full genomic information. So this is possible in this case. And this, of course, was quite interesting because annotating genome, deciphering reactions, doing the mathematical conversion, and the guessing at phenotypic states is something that's possible for other processes than metabolism. And this led us to lay out this vision in 2003 when we knew these models could represent certain types of metabolic phenomena, that the same could be applied to other cellular processes as long as we had the right information. So we could do this in principle for protein synthesis, metabolism, regulatory networks, and so forth. And if we did, we actually had a mechanistic framework for which to do multi-omic data analysis, which now represents one of the key challenges in biology. Very pressing issue that the NIH calls big data to knowledge, the big data to knowledge challenge. So this was a vision that was laid down in 2003, and Jenny Reed, who was here and on the faculty, was a part of laying that out. And there we are at the light moment, but to do this, I guess you needed to do some magic and be a cowboy, I suppose. So this vision has been realized now, partially, as I'll show you, and is leading to very fundamental results about cells, their behavior, and their properties. So it's a difficult process, and it takes a long time. And I'm going to show you how we have been able to achieve this for synthesis of the proteome, where we can calculate its composition, its optimal composition, and its balanced function to produce phenotypic states. So to do this, we have to reconstruct, just like for metabolism, all the processes that underlie protein synthesis. Fortunately, in enterobacteria, these are quite well known. And this involves understanding the RNA polymerase that transcribes the genes into messenger RNA. These are then translated on the ribosome, a very complicated operation, into the protein. The protein then needs to assemble into multimeric complexes. And very importantly, many of them do not function without the prosthetic group being engrafted in them, say like B12. So you can make the protein, but if there's no B12 in it, it doesn't function. And so these are metabolic products. In addition, there's a lot of modification of these molecules, like the tRNAs and alike. This is no small feat to do this. In E. coli, protein synthesis relies on about 400 gene products, many of which are not protein, but RNA-type molecules. So this now leads to different types of models, which I'm going to be talking about. There are the metabolic models that describe metabolic flows, as I talked about. But if you put protein expression in here, you actually also compute the proteins that are needed to carry out the reaction. So you can actually calculate the proteome here. So the way we think about this is that M represents, the metabolic flows. This actually represents the operating cost of a cell, because you got to make energy and materials, and these are the operating costs to run a phenotype. On the other hand, the proteome is capital investment, so that's like a capex part of running a cell. You have to invest before you can run the state of the network. If this was a traffic network, these were the flows through different streets and junctions, and this was building bridges and the roads and alike. So we now have basically mixed OpEx and CapEx models of cells. And this is leading to kind of a global econometric view of a cell and how to meet its physiological challenges. This particular feat was achieved by Ines Thiele, who is now at the University of Luxembourg. Now, to do this constraint-based calculation, we have to figure out what constraints the proteome is operating under. And these are pretty obvious in principle, but may be hard to cast into equations to get all the right numbers. So first, of course, is the turnover rate, and this can be estimated by a variety of methods, so we can get the turnover rates. We also have to balance the amount of every enzyme in the system, and so it has to be synthesized, it degrades, it dilutes by growth, and so you have a balance on every protein in there. But then perhaps most importantly, in the cubic micron, you can only fit so many protein molecules. If you can only make 2.5 million protein molecules to run a cell, if you're going to use 250,000 for the allocation factor, well, only you have a limited space for the rest. So you have to allocate the proteome optimally or suitably for survival of a cell. So I'm not going to bother you with any of the equations or how this is actually done, but it turns out you can build these complete models. And this leads us to be able to address some very fundamental biological questions that I'm going to actually focus on now. We're going to talk about here a little bit about the determinants of microbial growth rates, how we can actually figure out what limits growth. And more importantly, we're going to talk about how evolution or distal causation can now be studied in a laboratory, and we can figure out how phenotypes change under selection pressure. And there are many other questions we can answer on that list by being able to compute proteome allocations. And this work was done by Teddy O'Brien, who is now with Ceres Therapeutics, I think it's called, in Boston. There's a microbiome company that is treating C. difficile infections by using probiotics. It's been a very successful company. So let's now talk a little bit about evolution in the laboratory. This turns out to be a very fun thing to do. We can now actually practice in laboratories evolution of cells under controlled conditions. You can do this manually, which is difficult, it's very laborious, takes a long time. We've been able to automate this process under the leadership of Adam Feist by building an evolution machine. There is a picture of an evolution machine. It will carry out an evolution of bacteria in about 30 to 40 days. It can multiplex a lot of cultures, and it passes them with exquisite accuracy. And in fact, the evolutions turn out to be reproducible. You have to do a certain number of them because they are never identical, but they turn out to be now, what should I say, evolution can now be addressed quantitatively in the laboratory. And this is the type of results that you get from doing this. And here's a progress curve of improved growth rate. So this is growth rate selection. And the ordinate here, the time is measured in the cumulative number of individual cell divisions. And this is done because mutation presumably happens during division, and the likelihood of a mutation occurring, beneficial one, is proportional to the number of cell divisions. And we see progress like this interrupted by these shelves here. And so what's happening is a beneficial mutation appears here, fixes in the population, and then it reaches the benefit level of that mutation. Then there's a bunch of new mutations that come up here, and another one is picked, you reach another shelf, and so on. You can now resequence the cells after the evolution on these shelves and in the beginning, and you can figure out which mutations are responsible for these changes in fitness. And we can do that by editing the genome here that we started out with. You can just put the mutations in there and assess their causality. And that first jump is very often due to mutations in RNA polymerase. And some of them are shown here. These are single mutations. They're not quite unique, but there's a discrete number of them, that if you introduce them into the wild-type cell, its growth shown here in red, these are single mutations introduced, you get a much faster growth. Not only you get a much faster growth, but you get better biomass yields from introducing a single mutation. And this is actually a remarkable result, and some of the underlying mechanisms have been elucidated, and I'm not going to detail them here, but just to show you, maybe give you a flavor of how this is done. I mentioned that we can do molecular profiling of cells in detail. We can, for instance, profile all the population of messenger RNA molecules. And that's what's shown here for these two mutants, and each set is normalized to the starting strain. So 0 means no change in expression, and those genes are not shown in here. But there are two groups of genes that change markedly in both these mutants, shown in light blue. These are overexpressed, and those in light brown here are downregulated. Now we can look at the annotation of these genes and look at their function. And it turns out that those in brown are what are collectively known as hedging functions. These are stress functions, like response to pH, response to antibiotics, multi-drug resistance pumps and stuff like that the cell, protein that the cell makes but may not need, but it just makes them just in case they need them. So they're willing to invest in a piece of the proteome just to be ready for something unknown. The blue functions here, the genes in light blue, are growth-associated functions or growth-promoting functions. So you see a systematic reallocation of the expressed genes here away from these hedging functions or these stress functions to growth functions. And we call this the fear-greed trade-off. Cells becomes greedy when you evolve it in this constant environment where it wants to learn how to grow faster and faster and faster, because that's what the selection pressure is. And it becomes less cautious because it downregulates all these proteins it doesn't need, because after generation and generation in there, it learns it doesn't need them. So we can compute this, and the pie chart that I showed you before is here. We cannot compute the entire proteome. We compute about 80% of it by mass now. And that's this blue segment here. So those tend to go up, and the non-V proteome, the stress functions that are not yet modeled, tend to go down. We can also, from physiological data, estimate the operating cost of the cell. And we can put this into an engineering type diagram where we plot CapEx versus OpEx. So this is basically the cost of, this is the proteome allocation here, the efficiency of it, and this is the cost of running the phenotype. And this is the wild type. These are lines here, isoclines, lines of constant growth rate. The wild type moves to this point here, from here to here, just by introducing the single mutation. Both saving on CapEx and OpEx, in other words. Now, turns out that the genotype-phenotype relationship is multiscale, and we can now parse it out with all these methods that I mentioned and studied in detail. You are able to track out the causation of this mutation through all these layers, and that's illustrated here. So you sequence, find the mutations, determine their causality. You then look at the mutated gene product, in this case, RNA polymerase. And it turns out that these mutations impact the PPG-PP binding site. That is a ligand that's a metabolite that binds to RNA polymerase, and it's like a brake on it. It's an alarm signal. It'll just stop the function of the RNA polymerase quickly. So that is the putative mechanism at the protein structural level, not fully established yet. But we know in terms of the function of it, it wobbles the transcriptome away from these hedging functions to the growth functions. So this single mutation reprograms the transcriptional regulatory network. And it seems to come through differential use of sigma factors. For those of you who know this field well, this mutation not only affects the PPG-PP binding site, but seems to favor the binding of the housekeeping sigma factor, sigma 70, that tends to express these genes, and sigma S, the sigma factor associated with the stress functions, which tends to promote these. No mutations in anything else. It just tilts that balance. And then at the systems level, we can do what I just said. And then there's a number of phenotypic predictions made by this, not just the growth rate, but acid shock resistance and so forth. Now let's talk about this conceptually. So here is that progress curve that I showed you before with these three well-defined steps in it. Here is an image of that polytope that I showed you before. So this is a piece of that stoichiometrically defined solution space. And for a stoichiometrically optimal function of that cell, you should sit in that vertex, or this corner of the polytope. The wild-type cells aren't located there. There may be, there's a big solution space here. They may be close to it, but they are far away from it. They don't go all the way there because they have these fear functions. They have these hedging functions. We can now scale distances in this space here in genetic terms, in terms of mutations. For instance, this first progress towards that optima is a mutation in RNA polymerase. We know that this is related to nucleate-associated protein and organization of the genome. And this here, it rearranges a few things in the cell envelope. So we can now do this. Now we can understand what this cell is actually hedging against, why it's not growing at that optimum. And Vasily Hatzimarikakis at DPFL pointed out to me recently that this diagram I was showing was remarkably similar to that bull diagram I showed you before, that when you actually evolve cells, you can actually force them to the corner of that polytope. So now we remember that diagram very well because this was a very clairvoyant conceptual prediction. But let's talk about this broadly. We are now studying a bunch of different E. coli cells, and we are evolving them all into that stoichiometric optimality. And we are scaling that distance in genetic space for all of them. We are not so much interested in this state. We are interested in that state. And we are interested in actually rewinding these curves to figure out what makes one strain hedge differently than another. And this is very important in understanding strain-to-strain differences, how they choose to allocate the proteome, what they are hedging against, which in turn is presumably based on their evolutionary history. This is now being applied to starting strains for bioprocessing. There are about 7 popular ones, and we have now characterized and are characterizing in detail what really are the pros and cons between using these. We are also doing this for pathogenic strains. E. coli strains occupy different microenvironmental niches in the human body. Uropathogenic strains, for instance, infect the bladder. And about 70% of bladder infections in the US are by E. coli. They also populate other niches in the body where they are specialized to grow. And this here is a little classification tree of these different strains based on their metabolic capabilities. And you can see they classify based on certain substrate competencies. And these substrates happen to map onto the environmental niches where these cells populate. So it's very satisfying. And we are now looking at the proteomic allocation to see what they are hedging against in that environment, and what kind of virulence factors they actually express, and how much of them they have to express to induce a successful infection. And this has been done by John Monk, that is shown here, very happy after graduation. He's done this for a number of different pathogens, and this is actually remarkable, because this is done based on sequence information alone. All right, John, so what do I have left? 10 minutes or so? 4 or 5 minutes, okay.
Still allow for questions.
All right, we'll cruise right along. So this leads me to my second question here, which is what determines growth rate? We now have very deep proteomic coverage of different E. coli strains under many growth conditions. And we can now judge the efficacy of the expressed proteome relative to growth. And this summarizes those results. So it turns out, like I said, there are a lot of protein expressed in cells that are never used. So you grow E. coli on glucose minimal media, it'll have transporters for glycerol upregulated, even if there's no glycerol there. If you switch it to anaerobic conditions, it upregulates all the amino acid transporters, because it's expecting to be in your stomach now, where there should be plenty of amino acids. But they're not there in the media, so they make all this protein and they don't use it. Then there's also underutilized parts of the proteome, where there's an overexpression of the protein to carry out that physiological function. So we can segment the proteome into unutilized, underutilized, and optimally utilized proteome. And these 3 diagrams just show how the predictions of the growth rate improve as you strip these out. This is the whole proteome. This is when you take out the unused proteome. This is when you take out the underutilized proteome. Where you get a perfect prediction between the growth rate and the fraction of the proteome that is actually associated with growth. So these hedging functions are a huge determinant in the growth rate. So since I only have 5 minutes left, I'm not going to talk about this in detail, but you can computationally look at these different sectors of the proteome. There's a core proteome always expressed. There's a proteome, part of the me proteome here, that is expressed in a condition-dependent manner. And it maps perfectly on some known transcription factors for those functions, and this was published in PNAS last year. We can look at this composition in more detail. So here's the growth rate, and this is the percentage of this core proteome, as you would expect, goes up as the growth rate gets faster. Here is the C proteome, the carbon-utilizing proteome. That tends to go down as you grow faster and faster on a single substrate, as shown over here. And the non-me proteome has to be treated by bioinformatic means. Here are these functions, proteostasis, regulation, stresses, and so forth, that are not accounted for yet in this model. The abundance of these protein tends to go down with faster growth rate, but that's not true for all of these classes of protein, and the correlation coefficients are negative or positive, depending on whether these protein have growth functions or these stress functions that I spoke of earlier. So there are systematic changes in the proteome composition with growth. And the bottom line is this, that the proteome allocation has a lot to do with determining cellular physiology and the particular phenotypes that are expressed in a given environment. And this here shows some trade-offs in proteome allocation based on what I've said. So when you want to grow fast, you have to allocate a lot of the proteome to growth functions, but you have to let down your guard on stresses because you can't be prepared for them. But you also have what's called low substrate readiness. These cells, these evolved cells, for instance, to grow fast, can't go through deoxys easily. They can't shift from one substrate to the next because that C proteome is so small. It'll take them a while to upregulate that C proteome. But cells that are growing slowly, which most cells do naturally, have a lot of stress functions upregulated, and they are ready for many different environments in terms of substrates and so forth. So growth is actually a very risky proposition, as it turns out. Now, that's the end of the technical part of this talk. Say a few things about the future. Today, these me models calculate about 80% of the proteome by mass. We are steadily reconstructing more functions of a cell, like their location of proteins. We now have the 5 protein translocation pathways in E. coli accounted for. So we now allocate the proteome into cytoplasm, inner membrane, outer membrane, periplasm. and what is out hanging, sloughing off the outer membrane. We are also building the stressome. We now have information to build most of the stress function that I showed you. They account for about half of this, so we maybe soon will be calculating over 90% of the mass of the proteome. And we've now also been able to get the structures for each one of these proteins that allows us to study genome-scale models at an atomistic scale. So there's a lot of excitement still ahead here. It's okay, I'm done. So we are here to honor Ed Lightfoot. He's now 90 years old. He has touched our lives in many different ways, and here are some pictures of him and myself over time. The first picture I showed you, this is when I was a young faculty member at the University of Michigan. He came to give a series of lectures there. Here he is with us in 2003 in San Diego when that tissue engineering book was published and dedicated to him. I think I may end with one quick story of many I could tell you about my time with Ed Lightfoot. This happened during my time here in Madison in 2000 when I was here as a Haukan professor. And there was an election that fall in November, and I happened to be here on election day. And in fact, I was invited to Ed's house to watch the election results. And as you know, he has a dog that he likes to take out for a walk. So we are anxiously watching the election results. The results from Florida came in, Gore won, and he was going to be our next president. So Ed says, that's good, let's take the dog out for a walk. And he goes out for a walk, comes back half an hour later, oops, Florida recalled for Bush. Bush is going to be the next president. And needless to say, he was not too happy. But as you mentioned, a few years later, he had to go to the White House and get the presidential medal from President Bush. So happy birthday to Ed. Thanks to all of you for listening. Thank you.
We have allotted a few minutes for questions. So do we have some questions? Yes, please.
I guess most of your work was done with E. coli. My question then, if so, then how translatable would this be to other cells?
So a good question. So we've not only...
Maybe you could repeat the question.
Yes. So the results shown here are specific to E. coli, a well-studied organism. How does this translate to other cells? So we worked with many other enterobacteria, Salmonella, Klebsiella. We worked with Gram-positive, Staph aureus, and some serious pathogens. For the human system, a reconstruction of the global human metabolic map has taken place. It's been customized to different tissues based on expression profiling. And there's a growing number of studies associated with human metabolism. And there's a lot of effort now applying these methods to microbial communities for all the reasons that I mentioned during the talk. And this is all progressing pretty well. And I think this is a field that will be with us for a while and has a big future.
Jim Rawlings.
Interesting that your evolution experiments are reproducible. So why would you expect that, and what happens to the randomness?
So this is a great question.
Again, the question.
So the question is about the reproducibility of evolution. We view it kind of as a random process with unknown outcomes. I said it was reproducible. It's reproducible in the sense that when you evolve a bunch of parallel cultures at the same time, they tend to reach the same fitness state. But then when you sequence them and look at them, they all do it differently. So there are actually alternate optimal solutions to the evolutionary challenge problem. If you do this experiment over and over again, you see more variety, but they fall into certain groups. Like there will be a mutation in rpoB, but it may not always be identical, but it's going to be there. In some cases, there are magical mutations that have to be there. In others, there is slack. And in some cases, there are apparently very, very many ways to get to an optimum. So we have a narrow path to an optimum, and we have a broad path to an optimum. And we are now trying to study why that's the case. And you probably already know that mathematically, when you do constraints-based optimization, there are equivalent optimal solutions. So they're often an edge of a polytope or a whole surface. And we are now discovering that these certainly, those predictions are biologically relevant. So it's predictable at the overall phenotype level, but maybe not at the molecular profiling level. But seems to fall into categories, though. It's not an infinite number of choices.
Maybe I'll jump in on that. And in the first step, you mentioned mechanistically this was perhaps distributions of sigma factors for transcription. You also had a second step, though, and I wonder if you have some insights on the mechanisms for the second step of the evolution.
So we do know that the gene products that are mutating then are basically DNA binding protein, so-called NAPs, that determine the structure of the nucleoid, but they also open and close certain parts of the genome for expression, because they tend to bind in bands and often are also mistakenly called transcription factors because they simply block a gene as they're folding up the nucleoid. So there's something about an optimally efficient geometrical conformation of the nucleoid that happens.
So it's somehow a physical constraint in the DNA that's being
Yes.
enabled.
And probably you might guess that that may be a part of resistance to phages and try to mock up viral infections and stuff like that. And if you open up your genome fully, now you're probably much more susceptible to any viral infection.
How general is the notion that maximizing the growth rate should be a objective function for an organism?
Yeah, so that's a poor objective function in general because most organisms around are not doing that. The organisms we've been growing in the laboratory for a long time seem to have adapted that way. Bioprocessing certainly is focused on optimal growth rate and metabolic properties, so you can now use the adaptive evolution in the lab to try to reproduce these properties. And there's a lot of work now going on trying to define selection pressures, and you can do many more of them than growth rate. Well, you select on growth rate, but you can steer into one of these solutions that you're interested in that Jim asked about. But generally, so organisms out in nature are not trying to grow quickly. Their objective seems to more be to harvest rare elements like phosphate and nitrogen, for instance. Bacteria, out in the soil double twice a year or something like that. So people are trying to use many different objective functions to study these models, optimal ATP production in the mitochondrial model, and there are many objective functions that have some biological reasoning that we're trying to look at. The real objective function, like I said, can be studied based on forcing it into the optimum, which could be thermodynamically like delta G0 or something like that, but delta G, the actual one, is actually some distance away. And you could maybe now figure out why they are some distance away. That's what we are mostly excited about, is to try to determine what a wild-type cell chose to guard against and why it chose the physiological state that it implements under the conditions you put it in.
Other questions? Maybe I'll just ask one more. To what extent do you think we'll be able to engineer the consortia of bacteria that are determining, to a large extent, our health?
Well, as you know, there are many business plans you can do that. I don't think I have any particular insight into that. I will say, though, that the studies we have done on microbial consortia in my lab and that of collaborators is that consortia of microbes are tied together at 3 levels. They clearly exchange macronutrients, say, based on thermodynamic and stoichiometric considerations. Then they become willfully auxotrophic, and they exchange compounds like amino acids willfully, and they're tied together like that. They are mutually auxotrophic. And then there's a 3rd level that is quorum sensing and at that kind of level. And so when you evolve these communities or put them under different conditions, the population composition changes, but nobody's really wiped out. It seems to be a stable composition of the community, but changes in its composition with environments. So I don't know. We'll see. We should write grants on that.
Very insightful. So thanks. Let's thank our Bernhard once more.
Thank you.